Well, here we are for lecture 3 in this fourth module, and now we're getting to that plasma membrane that I keep bringing up again and again and again. And we're going to talk about the various characteristics. We're going to go through each one of these one at a time. We're going to talk about the phospholipid bilayer, which once again, by this point, I would think that you've got this pretty well down, that concept once again of my little phospholipid, and they're just facing each other, right? With a inside lipid tails, that fear water, hydrophobic, on the inside, and hydrophilic phosphate heads on the outside, along with occasionally embedded proteins. And what's important in this is, yes, the proteins are embedded. We've talked about that. We've talked about how this part of the protein that is facing the outside of the cell, and this part that's facing the inside, that's next to these the phosphates and the phospholipids, these would be more of those hydrophilic or charged amino acids, and these in the middle that are next to the lipids, well, they'd be more hydrophobic. But notice that they move in space. So this is not something like it's rigid. It's not rigid at all. It's very fluid. And it's very important to remember this entire membrane that covers each cell is not just solid. These molecules are moving fluid in space. And part of the system that makes it fluid is the molecule of cholesterol, which once again is another specialized type of lipid. We tend to think of the word cholesterol, especially in the fitness industry, oh, cholesterol, a bad thing. No, cholesterol is a very important molecule. It's a very important molecule for exactly this. Membrane fluidity is the term. But also, it's important for making lots of other structures, including hormones. Many hormones, what's known as your steroid hormones, all right, are made from cholesterol. So, important molecule. We'll talk about dietary cholesterol a little bit later. But for right now, I want you to think of cholesterol being a good thing, allowing for membrane fluidity, and also being the structural foundation for many of your hormones, specifically the steroid hormones. But wait, there's more. Look at this. It contains both proteins and carbohydrates and lipids. Oh my goodness. So we know that the phospholipid bilayer, by definition, is a lipid. And we know that there's proteins embedded in it, which are going to do lots of interesting jobs we're going to find out. Right? But then here's that other organic molecule, carbohydrates, that are going to be connected to many of these proteins and or many of these lipids, and have various functions as well. So I kept telling you in Chemistry of Life chapter that these organic molecules, there were 4 of them, carbohydrates being 1, proteins being 2, and lipids being 3, are going to visit us again and again and again. Of course, the 4th one being nucleic acids. Well, that's going to be your DNA and your RNA, but also, where it's going to even relate more to this part about the membrane, we're going to find out it's going to fuel active movement across this membrane. And how's it going to fuel it? ATP is a nucleic acid, your main energy molecule in your cells. So once again, hey, there's those 4 big organic molecules that I warned you would come back again and again and again, and here they are. And the other big important point that I want you to realize is that these membranes, these phospholipid bilayers, are selectively permeable, which means some things can get across the membranes and some things can't. And what's going to determine that? Well, it's going to be obviously whatever that molecule is that's trying to get across one side or the other, but also those proteins. So the cell, in a sense, decides which molecules, which ions are going to be able to get across and which ones can't. So here's a zoomed in artist rendering of that phospholipid bilayer. And here, once again, here's their image of that phosphate head, lipid tails facing each other. I think you guys have got that down by now. And here, once again, they've defined the hydrophobic tails, hydrophilic heads on the inside here, outside, extracellular out here, intracellular in here. What else do we see? Well, oh, we've got these big proteins. These big proteins that are spanning that phospholipid bilayer. What else do we have? Oh, we've got these 4 ring structures. That is cholesterol, which is going to help this fluid membrane be more fluid. Then we've got these yellow things. What is that? These chains of 6 rings. Oh, those are carbohydrates. Carbohydrate chains that are connected to either a protein or sometimes directly to the phospholipids. So once again, we should just get an idea of this membrane when we zoom into it, its basic structure, and realize that once again, it's not stagnant, it's not frozen here. All these things are moving together, but still maintaining the boundaries, the difference between the outside and the inside. So going back to this concept of selectively permeable, once again, the membranes allow some things to cross while keeping others out. So once again, we have in this little image, we've got the inside and we've got the outside. So some small molecules like water, and actually for the longest time, for years, they didn't know how water really got across, and water does. Water gets across freely, which doesn't make sense because it's so highly polar. And they used to say, in fact, one of the animations I'm going to show you still says, well, it's just so small it can get across. Well, that's not entirely true, because it's polar, it's small, but we also have smaller ions that can't get across. So now they think there's these pores called aquaporins that allow water to move absolutely freely from one side to another. However, we've also got potentially charged molecules and ions, right? Full charges on here that can't necessarily get across because they're, because of their charges. They can't get across this phospholipid bilayer unless they go through a specialized protein like this channel right here. Same thing, big macromolecules, they're not going to be able to get across this thing, whether it's charged or non-charged. The charged molecules and the polar molecules might get up to these polar heads and like, oh, we'll be friends, but then it can't get through the middle part that is hydrophobic. And exactly the opposite here, these big macromolecules, especially if they're non-charged, non-polar molecules, they might come up to right here, but these hydrophilic heads will repel them. So some things can sneak through that are small enough and go through specialized pores, but for most part they're separated, and the only way they'll get across is by those membrane proteins. So how do they move across the plasma membrane? Well, there's 5 different ways something can get across the plasma membrane, and we're going to cover each one of these. There is diffusion. Now, just basic diffusion. What is diffusion? Going from a high concentration to a low concentration. And what's really happening here with diffusion? We've all seen this, in our basic science classes. A classic example is you take a cup of water and you put 1 drop of food coloring in it. And of course, you see this very intense color at first in that droplet, and then all of a sudden it just diffuses throughout the water and separates and spreads out eventually evenly. And the concept here is you have to remember molecules move and there's energy in them, and they start bumping around each other, and because of this bumping they start moving around and around and around, and they start separating out more and more and more. The more energy in a molecule added to a molecule, for example in the form of heat, the more bumping that's going to happen. So molecules are always bumping into each other, and they're going to just kind of spread out and increase in entropy and increase in chaos. So that's what's happening. We go from a high concentration of something, like say you're driving down the road and all of a sudden you see a skunk in the road and it sprays, and it comes out of its back end, and all of a sudden that scent is going to diffuse through the air very, very quickly and of course stink up your car. Same concept, right? Now, osmosis is a special form of diffusion of water across a semi-permeable membrane. Once again, semi-permeable meaning is only permeable to some things. It's like selectively permeable. Selectively is permeable. We use that word selectively because we have selective protein channels that are going to let some things versus others. Semi-permeable just means there's not that protein control. Some things get across, other things can't. Well, if you have this semi-permeable membrane and you have a high concentration of a solute on one side and low concentration on the other, the water is going to want to flow across to try and even it out. And we'll see that a little bit more in a second. Facilitated transport, that's that use of protein channels across the membrane. But the important thing here is that there's no energy involved. In other words, there's no use of ATP. And these molecules that are being transported through these specialized protein channels, they're going down their concentration gradient. So it's still a form of diffusion, if you will, but it's through these facilitated transport. Active transport, on the other hand, still protein channels, all right, but now energy is required. So it means we're going to be using ATP. And often, with active transport, they're going against their concentration gradient. So we're actually pulling the molecules from one side to the other. Lastly, that's everything we've talked about so far. These first four are about small molecules. We're about the big molecules, the real big proteins, big globular proteins or lipids or what have you. Well, then, in order to get it into the cell, we're actually going to invaginate that cell membrane, grab it at the cell membrane, and do what's called endocytosis. Or if we're going to release something outside the cell, say we've actually translated a protein and we wanted to get it to the outside of the cell, maybe it's a hormone, for example. Well, it'll go all the way to the cell membrane to be packaged in these vesicles, and that vesicle will become one with the cell membrane, and that's exocytosis. So this is endo, okay, cytosis, the cell, and think movement, okay. Exo, outside the cell. Cyto, so cell. Okay, so let's look at each one of these. So once again, diffusion is the random movement of molecules from a higher concentration and from a lower concentration. And we're looking at, once again, the difference between diffusion and osmosis. So osmosis is specifically the diffusion of water molecules. That's the important thing with osmosis, is that it's all about H2O. Okay? So over here, once again, we have a plasma membrane, and we have particles on the inside, and we have the same particles on the outside, and the little red and white here, that represents water. Now notice that comparatively the ratio of these particles to water is a lot greater over here than it is over here. Now particles aren't necessarily going to be able to get across, but the water can. And so the water is going to want to try and flow out, so it's more or less an I'm going to say equilibrium, that's the word I'm searching for, equilibrium condition of particles going in and out. So let's give you a little example. It's an interactive thing, so we're going to begin. And here we have, first of all, in this little beaker here, extracellular fluid with membranes permeating substance, all right? And here we have a suspension of cells. So just in this little dropper here, we have a fluid with a whole bunch of cells in it. So I'm going to drop in a drop of cells, and we're going to zoom in and look at that cell, and here's that plasma membrane, and here's the nucleus, and this nuclear envelope is nuclear membrane. And we have out here's water, and the purple is, once again, that solvent that's in the water that actually can get across. So now we're just talking diffusion right now. What's going to happen is right here is the concentration of the solute in that extracellular medium. And here we're measuring the concentration of the intracellular. And what we want is that equilibrium. We want equal numbers, more once again proportionally, if you will, concentration, once again proportional, of these little solvents on the outside to the inside. And notice, because the extracellular is so much greater, we don't actually see this concentration go down, even though some ends up into the intracellular area right here. So this is constant, but eventually what's going to happen is that those particles, if they are allowed to diffuse across passively, it's going to be this even concentration of the solute within the extracellular medium and the intracellular medium in the cytoplasm. So same concentration. All right.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay, these last two words given here, the hypotonic and hypertonic, are actually going to be very important when we talk about the cell. So let's look at these terms again. So once again, isotonic, and what we're talking about here is a concept known as tonicity. And it has to do, once again, with the solute concentrations on either side of that semi-permeable membrane. So if I say iso, meaning same, they have equal amounts of solute as far as the concentration inside and outside, so water doesn't flow in and it doesn't flow out. If you remember in that example with the beaker, we actually saw so much water flowing from one side, and I'm not a very good artist here, but here's my beaker. We saw that water flowing from one side to another, that the levels actually changed, and you can imagine how there's actually force being created here, and because of this flow of water from one side to the other. And that's what happens when we have unequal solute concentrations, such as a hypotonic solution. So when we're talking about hypo versus hypertonic, we're first of all talking about the solutions. We're talking about, let's say, what's in the beaker itself. And hypo meaning low, and hyper meaning high. We're going to come up with those prefixes again and again and again. And what can happen here is if there is low solute concentration here and high concentration here, the water is going to be flowing into, as you can see from the arrows that the publisher actually gave, the black arrows here, going to go into the cell. And it can actually go so much into the cell that the cells will burst or lyse. Okay, lysis, bursting. So we're going to see this is a very common word and a term often used as a suffix in biology. An example just as outside of this would be like glycolysis. Glyco meaning relating to sugar, lysis once again meaning breakdown. So this is our process of breaking down sugar for energy. Only reason I bring that up is so you understand all these words are going to come back again. And these terms, so prefixes hypo and hyper, very important. Lysis as a word itself, meaning bursting, or as a suffix, we're going to hear again and again. Back to the original point, once again, hypotonic solution, the water is going to flow out of the solution and into the cells, perhaps even making them burst, lyse. What if it's the reverse? What if we have a hypertonic solution? Well, then the cells are going to shrivel up because the water from inside the cells are going to want to go out in the solution to once again try to make it as much as it can isotonic. And once again, because that solute can't get across, the water is going to flow out of the cell. And because, once again, in this case, the solution has more of that solute out all through here, water is going to flow out, and the cells are going to shrivel and shrink. Crenation. All right. Well, we talked about diffusion, we talked about osmosis, and we introduced the concept of tonicity. Now we're going to talk about facilitated diffusion and active transport. Now, I already mentioned facilitated diffusion, one, has to take part at a special molecule that crosses that membrane, a specialized protein channel, right? And it's going to be going from a high concentration to a lower concentration. So we have something on the outside, for example here, that's in high concentration, in this case it's glucose in our example, that is going to go from a high concentration out here to a low concentration. Is going to be naturally pulled in, but if it weren't for this protein right here, this special protein channel, this protein carrier, there's no way for it to get across. So it's going with its natural concentration gradient and no extra energy is involved. Well, in other words, no ATP. So let's take a look at that.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Well, that was facilitated diffusion. Now let's talk about active transport. So active, as the name implies, it requires energy, and in fact it actually pulls things against their natural concentration gradient. And one of the primary examples that, once again, I use the term frequently, that you will see again and again and again, is called the sodium-potassium pump. And we're definitely going to see this when we talk more in anatomy and physiology about the nervous system, because this is what's going to actually fuel, in a way, the nervous system to allow impulses to be sent down neurons, those nerve cells, those nervous system cells. So, in this case, once again, we have, as an example, sodium here on the outside, and there's a higher concentration out here, and on the inside, lower concentration of sodium. And what's going to happen is they're actually still going to be being pulled out against their concentration gradient. And once again, here's my magic little molecule here at the bottom, ATP, that's going to fuel this whole thing. And at the same time, while 3 sodiums are going against their concentration gradient, 2 potassiums are going to be pulled in. So once again, sodium represented by Na ions, their positive ions, are going to be switched out for these potassium represented by K ions. It's always a 3-to-2 ratio. And what that's going to do is, once again, they're going to drive against their concentration gradients, and it's also going to create a net charge difference, a polarization of charge on either side of the membrane. So let's take a look at that.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, lastly, we're going to talk about how do we get big molecules inside and outside of the membrane. And that's endocytosis and exocytosis. So endocytosis transports molecules or cells into the cell via an invagination of the plasma membrane to form a vesicle. So now we're talking once again going inside the cell, pulling things inside. Whereas, of course, exocytosis transports molecules outside the cell via fusion of a vesicle with the plasma membrane. So looking over here, here is phagocytosis, and here we have the plasma membrane, here's the outside, and we're going to have this, say, this big globular protein thing here. And it's going to come right up to the membrane. The membrane is going to what's called invaginate. So it's going to kind of come in like this. And eventually it's going to pinch in. That's really what's happening. It's going to pinch together and then separate. So now we have a new membrane-bound vacuole holding what the cell wanted to bring in. So a piece of the outer plasma membrane pinches in and grabs hold of whatever this big molecule that the cell is trying to bring in. Same idea here is that's phagocytosis. These are large molecules. Sorry, there we go. And penocytosis is a whole bunch of smaller molecules brought in kind of the same method of an invagination, pinching in. It's just what we're bringing in are just smaller. So phagocytosis is large, penocytosis is small. And then there are times when we have receptor-mediated endocytosis, where these molecules are going to be binding those protein receptors, and then the cell invaginates, and that's how they make it to the inside of the cell. Right? Now, exocytosis, imagine the exact opposite. Say we need this, I'm going to look at this big picture up on top again, and say we want to release this big molecule to the outside. Really, it's the exact same thing in reverse. I'm going to turn this arrow around, and this vacuole is going to come up to that plasma membrane and become one with it, and it's going to release and open up and release its contents that was on the inside here. It's going to release it to the extracellular medium. So once again, one last little animation from the publisher.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the.
Well, I don't know about you guys, but personally I tend to be a very visual learner. So I hope these animations help. So you can visualize that the endocytosis, whether it be a phagocytosis where it's a large molecule, often it can be some sort of disease-causing agent like a pathogen or something like that, or whether it's just more of a liquid and small molecules like pinocytosis. Hopefully you can get a better idea watching this animation how the cell just pinches, invaginates and then pinches in and actually creates that new membrane-bound vessel, bringing in what was once on the outside, and then once again that reverse movement and going on the outside. So I hope these animations, I think they're pretty decent for the publisher, help you visualize these things. That's it for the lectures for this week. It's a little bit shorter than, say, last week overall, but I really need you to get these concepts down because once again we're just going to keep building and building and building on these key concepts within last couple of weeks of chemistry, and now this week and next week in the cell. Everything is going to be setting on these foundations. So once again, hopefully these visuals helped, and I'll talk to you next week.